Okay, I guess with that I can start, right? Hello everyone, I'm René Inckemann, and I'm actually working with algae, so I'm working with Chlamydomonas, and as I said, I'm also supervising the iGEM team in Marburg, but in my PhD I'm working with Chlamydomonas to engineer the chloroplast. And with that, I would like to ask the other ones, which maybe not the ones, but from Marburg, but like, what are you working on? Which team are you from? What about Jared? Jared? What is your team? If you have a mic, I don't know. You can also write in the chat. Or like someone, huh?
We're on the same team. We're from Arizona State University in the US. We're also working with Chlamydomonas.
Ah, nice. What about Joel? Also the same team, or?
Yeah, I'm in the same team as well.
Nice. So we have one Chlammy team here. Nice. Is there actually, do you know, like, is there any other Chlammy team in the competition this year? Or are you the only Chlammy team?
We have no idea. This is our first year competing, so
okay,
trying to figure everything out. Yeah.
Then this is odd, right?
I think Sorbonne is working again with Chlammy. And also Nantes in France is working, and like I'm not sure. University Southwest in Australia seems to be working with Chlammy, and like two Chinese team I know of, like DUT China and SCAU China.
I think we should make this information somehow available or ask the teams, because that's a great resource to connect with other teams working on the same organism. I don't know if this is possible because of data protection, but like I would like to see this like connection going on. Okay, with that, knowing that there's one Chlammy team in here, I'm very happy that I have some audience here. So I don't know if you have any experience working with this algae, and we can do this more interactive than a standard talk here. So feel free to see this as a discussion and not like a normal presentation. And if you have any questions, also feel free to interrupt me, and I will try my best to help you out. So with that, I will start. So of course, there's not just Chlammy, but there's different algae, a lot of different algae, but there's not too many where some tools are developed or where people working actually in the field of synthetic biology with it. So there's Chlammy, as I said, then there's Pheodactylum tricornitum, which is a diatom, which is quite famous, and there's quite some tools for it. Then there's Nanochloropsis and some Chlorella or some other, but as I said, like maybe a handful of different chassis, but the best studied, as you already chose, the right one is definitely Chlammy. So there's no doubt that any other can compete with the number of tools. So the workers for algae biotechnology and synthetic biology is definitely Chlamydomonas, as I said, and therefore it's also sometimes called the green yeast. And actually, you cannot like engineer just the nuclear genome of Chlammy, but all three genomes, meaning like the nuclear one, the chloroplast one, and the mitochondrial one could be engineered theoretically. But I will come back to that later. Also, what is also quite special about Chlammy, it can not just grow photosynthetically, but you can also grow it on acetate, which means even if you grow it in the dark or have like a mutant which is lacking some photosynthetic gene, can still grow, which makes it very useful to study photosynthesis, also for engineering. And as I said, many tools are available. So how do you actually transform Chlammy? I think the most used one, or the most efficient one, is definitely electroporation, right? I don't know what you guys are trying to use, but that's definitely the most used one for CLAMI for nuclear transformation. And I will also mention some details afterwards, what is very important while engineering your strain with electroporation. But let me first introduce some other techniques, and that's, for example, glass beads. Glass beads are, on the one hand, you need way less equipment for it. You basically need just some beads, and then you put your plasmid or DNA in, and then vortex, and the DNA gets in, but it's way less efficient. So from my experience, although there's some other reports, you just get 1 or 2 colonies with that vortexing with glass bead methods, but like hundreds or thousands of colonies with electroporation. So if you have the equipment for electroporation, definitely go for electroporation. If you really lack the equipment, you can maybe give glass beads a try. So what's also not very clear in many papers where like the transformation is described, and if you have no one really showing you the procedure, or have any CLAMI expert in your lab, or like any algae experts in general, but the recovery step after the electroporation is very, very critical. So this is the point where you can fuck up, and if you are very unlucky, do not get any transformants. And you really have to follow the exact steps in the protocol with the low light, the exact shaking, and the buffers and the sucrose, because a slight change in that, for example, a colleague of mine, he just like did this transformation and thought, okay, my light intensity is not perfect, I will just put it in the normal incubator. And after a few days, he just saw like everything died. So like following that recovery protocol after the electroporation, but also on the glass beads, I think, is very, very critical. So if you have any struggles with the transformation protocol, that's the first step I would look into. Yeah, if you have any problems. Then the next problem with nuclear transformation in general in algae is that, as we discussed in the end of the talk, as one of the questions also was, that the integration is random in the nuclear genome, which means that you have very, very high positioning effects and also like silencing effects. And you would need to screen a lot of different colonies to find a colony which is actually expressing your transgene in a proper amount. And on the one hand, also if you want to verify it genetically with colony PCR, for example, that's also not as easy as in E. coli. And there I would highly recommend also looking into some published protocols. There's like some nice papers which came out just quite recent on like how to optimize your colony PCR in CLAMI. So it seems very trivial. It is not. So even me and like my students have been struggling a bit on like getting proper colony PCR bars running in CLAMI. So also there, it's really critical to follow the protocol quite exactly. Otherwise, there's no colony PCR at all working. The next thing is that people also have to struggle not to genetically find the right transformers, but also like the ones which are expressing, because depending on the position, as I said, you get a lot of different amounts of protein expression. And what people tend to do in the recent years is coupling the transgene, for example, to a fluorescent protein, and then like screening for fluorescent signal, either with like fax. If you don't know it, it's not important to know what it is exactly, or like just a plate reader or like fluorescent measurement, that even if it's not a fluorescent protein you want to produce but an enzyme, they fuse it together to have an easier assay to see if CLAMI is expressing your transgenes. And some people even got this working on like full plates. I saw a group in Bielefeld actually doing it with a camera system, which can like measure the fluorescence on an agar plate in a very, very high throughput. But this is probably not feasible for an iGEM team, but like the general procedure to link your gene of interest to something you can measure more easily than an enzyme is generally something I would recommend. There's also other systems. For example, I saw another iGEM team from Sorbonne, I think 2019, establishing a luciferase-based system for that. So you might need to, or might want to look into like screening methods for this kind of procedure. If you're not working with CLAMI, which I think no one is in the room, but there is some methods for example, pre-triconitum, where you can like get your genetic material into the diatom or the algae via conjugation with E. coli. I think some people tried it, for example, with agrobacterium in CLAMI, but not with E. coli. I haven't seen anything like that. But that's also something very interesting, in my opinion, that you could use E. coli here to get more efficient transformation by a conjugation. But as most of people are interested in CLAMI, I will just go over this very quickly. And I would just want to highlight two very specific tools for CLAMI here again, which might make your life easier working with expressing anything in Chlamydomonas. And one thing, which also the Kaiserslautern team mentioned in the beginning, is that there is like a very extensive modular cloning toolbox for Chlamydomonas for assembling your plasmids in a very easy way, in a very modular way. And you can just get these plasmids from was it Edging or CLAMI collection? So there's a way to order these plasmids, and then you can just start with your projects way easier because you already have all these promoters, these 5' UTRs, these introns, and so on to work with as a starting point. The second thing I want to highlight, and that's something my colleague in my lab also found, that there's a huge difference, is that like CLAMI is an eukaryote and it has introns, and it has been found in not just this figure in this publication, but in many other publications, that if you really want to have high expression, it's very, very highly recommended to use introns in your transgenes. And for example, you can see like the difference between no intron and using introns also on the like number of colonies you get. So if you design your genes, you could always already include the introns. And to make it a little bit easier, there's actually a website I would like to recommend, which is called Intronserter. You can just Google it and find it, and it will automatically place some introns in your gene at the right spot with the right sequence, and you can just order it in that way after using that tool. And this would already increase your expression because using these introns. So now I've talked a lot about nuclear engineering of CLAMI, and as I said, there's a lot of challenges with like the gene expression level, with the positioning effect, with the gene silencing. But there's also a method which has not been used extensively in iGEM, but I still want to mention it here as a method because I think it's feasible, would be feasible in iGEM, and yeah. So and that is chloroplast transformation. So CLAMI has one big chloroplast which has many genome copies of its chloroplast genome, and there's like a few methods available to transform CLAMI's chloroplast genome. And one is the gene gang, where you would actually shoot gold particles which contain your DNA on it, and shoot it on the cells to integrate. course, you would say now, which lab has this gene gun, especially as an iGEM lab. But there's also this method with a glass bead vortexing, and I did it myself. And it's possible even to transform the chloroplast with this glass bead method, although you still get just 1 or 2 colonies per transformation. But I would say it's still feasible for an iGEM team. And the way it works is that after chloroplast transformation, you just have one of the copies transformed, right? And then you would need to keep selecting the chloroplast transformation. And by keeping the selection pressure high, you would increase until you reach the point where all the chloroplast transformation copies are transformed. And the advantage of chloroplast transformation is that you, on the one hand, can choose the integration site by having homologous sequences. And also you get very, very high and reproducible gene expression by that. On the other hand, it's quite challenging to engineer the chloroplast, also from the chloroplast transformation procedure and the time. But still, you can have this in mind maybe for your project, design of the project, as an option at least. Yeah, here I just mentioned that glass beads are an option for chloroplast transformation when you don't have access to an actual gene gun. And with that, I would like to start a discussion on maybe what your project is about and where you are at. Oops. And yeah, so I'm happy to take any questions and also start the discussion with you. There's a question from Finn, I see.
Yeah, I just have like one little question. I wondered, why actually is it that you can't use electroporation to engineer the chloroplast of like a clammy?
Oh, that's a good question. And I'm not sure if I can answer the question, but just from a logical point, to reach the chloroplast, it's another 2 membranes you have to pass, right? Not just the cell wall, the cell membrane, but also then 2 chloroplast membranes. And I think from the way electroporation works with disrupting the membrane potential and disrupting the membranes, probably you don't get through. But I'm not sure, maybe it's possible and no one tried yet. So yeah, but that's not at least the standard methods you would use for engineering clammy in the chloroplast. So maybe about the ASU team, where are you at, and do you have access to the lab at the moment or for this year? And yeah, what's your project?
We do. We got started in the lab 2 days ago, pretty much. Nice. And we do have access to a gene gun. We're going to use that for chloroplast transformation.
Oh, really?
Yeah.
Super cool. Yes. Even the team which is working on the chloroplast. So what is your project about, actually?
Yeah, we're going to be kind of using clammy to uptake arsenic and reduce it to a soluble form. And then we want to build a filter out of it. But we're trying to do it in the chloroplast. So
nice. So would you like to introduce the arsenic in the chloroplast, or how would it work?
Yeah, the genes are going to be expressed in the chloroplast. And then, from there, I don't think they're secreted.
Yeah. So do you have actually a PI or someone working in your lab working with chloroplast transformation, or?
Yeah, Dr. Kevin Redding. I don't know. He's maybe,
Nice. So there's some expertise helping you on how to do a chloroplast transformation.
Yes, he built his own gene gun, so we're going to be using.
Wow, nice. That's actually a real thing in the field, that there's actually just one company building these gene guns commercially, and then all the consumables are super expensive, and this is really inhibiting the whole field, or young researchers working on this, because the gene gun itself, it's I think 30 or 40K or $40,000, which is insane for this stupid device. So I can totally understand that your PI or like your whatever it is building his own thing on, because this is really a limiting thing. So nice that you have access and that he is really pushing this.
Yeah, we're very grateful.
Yeah, but sounds like a cool project. So if you need any help, you can also always contact me, as this is my basically my PhD project, engineering the chloroplast of Clammy. So I know you have already quite some help. So yeah. So if you need any more help or any more advice, or if you need like a specific genetic construct or like anything like that,
Absolutely.
always feel free to contact me.
So is anyone else working with Clammy, or is it just us in here?
So we do, but not anymore as an iGEM project. So not really.
More professional right now, right?
We try.
Yeah, we've definitely been going back to your wiki from 2019 and looking through what your project did.
Yeah, if you have any question, you can always reach us, and also our PIs are really supportive for the iGEM spirit. So maybe if you're searching someone for your gold criterion, there's something like asking expert and getting using his feedback to improve your project. If you plan to do something like this and still search an expert, maybe you can ask for Michael Schroeder. He supported us and other iGEM teams really well.
Fantastic, thank you.
Yeah.
René, I think you were looking for a different transformation method of the chloroplast already some time ago. Do you want to update your findings or your results?
So I actually got access to this material. So the question from Adrian is going this direction, that there was a publication on like using carbon nanotubes for chloroplast transformation on plants. And I was interested in like, can you use that for Clammy? And actually they haven't even transformed the plants, but just got DNA in there. And like, I got in contact with that group, and they actually, after a lot of Corona problems, were able to send me the plasmids this January. So it took like another year to get the hands on this material. And I just tried it actually 1 or 2 times. And in these cases, I didn't receive any chloroplast transformation, but there's like such a big space of things to vary in this protocol. And I just didn't have enough time to invest in this project. But I still think if you would spend a whole PhD on that, you would probably be successful. But I got some other projects from my PI, and now I have to focus on that. That's why I haven't got any nice results. But yeah, it's still like a huge thing that it's not very high throughput to use the gun. Yeah, and therefore it would be super nice if there would be any like transformation method where you just pipe the DNA on and then the chloroplast is transformed. But this is still in the far future, I would say.
And
yeah,
another question: if you transform the nucleus with glass beads, are you only getting 1 or 2 colonies per plate, or because we have much higher success rates, but also we now switch to electropurification because the transformation of the chloroplast, and also because the only reason we are still working with glass beads is because I think that's a thing the Sorbonne University does, and Michael is coming from there, and it's his.
Yeah. So like, what was your what was your success rate with the glass beads? Just the estimate.
Verifying, but I think between the 5 and 100 or 200 colonies per plate.
Not bad.
And also, our regeneration protocol is not so problematic, or you don't have to follow it this much as with the electropurification. If I got it right.
Yeah.
We still also, because of Corona, couldn't do a electropurification myself, but I hope I can do this in the future.
Yeah. Yeah, for me, or at least in our lab, this is super critical, like following this recovery. But also after chloroplast transformation, also for you guys, like there's this step where you put clammy in the dark. If you do that too short or too long, they will just die. So really keep that in mind, and don't forget the plates, as I did several times after the whole procedure in the dark, and just remember 3 days later, fuck, I forgot the plates. No, but like this regeneration after any transformation of clammy seems to be very, very crucial.
Yes.
I don't know, for the glass beads, you must have like a super good protocol that this really works so nicely for you. But yeah.
I actually have another question in general. So I wondered, isn't there like there's probably some kind of mechanism to transport vesicles in the cell towards the chloroplast, right?
Yeah.
Isn't there like a possibility that you could like use this mechanism and like insert a gene into a vesicle, and that the cell absorb, like take the vesicle just to the chloroplast without actually like damaging the cell?
So you mean like, but on the other hand, then you would need to get like the vesicle into the cell.
Yeah,
Or
but
do you
I mean, there are like, I mean, if you already have a vesicle, there are like quite a number of like options. I mean, it's a bit different with plants, I guess, but for a normal cell, I would think that there are like enough receptors to just like include the vesicle.
Yeah, but still you need to get like a charged molecule of DNA into like the cell, and that's already like a charged molecule of DNA, which is quite big, where non-charged or like membrane is not easy. So that's the problem, I think.
Yeah.
And then like another few membranes to the chloroplast, that's very challenging. And the like the nanoparticles, which I was trying or like talking about, they work actually like that they are kind of compensating the charge of the DNA a bit by winding the DNA around like this tube of carbon. Yeah, but interesting question still. Yeah. It's still not like E. coli to transform the chloroplast, or like in general clammy. It's not as easy as it looks on paper sometimes.
Do you think we can combine the DNA then with histones and also get better transformation results, because they are also heavily positively charged and seems to be the thing in nature which takes the role of this nanostructure?
Are there even histones in the genome of like a chloroplast? I don't think so, right?
No, not in the chloroplast, but in the nucleus of plants and algae there.
Yeah, but that would be a huge problem if you have like the DNA wrapped around an histone. You need like there are mechanisms, of course, to unwrap all that, but if you do it in the chloroplast, I could imagine that the chloroplast just can't do it.
Yeah.
Yeah. Okay. Is there any like more specific question to like algae, or like what would you like to learn, or like where would you need to have help maybe in the future, or yeah, is there anything? I'm actually really excited to hear about all the projects of all the people participating. And I think I just saw in the chat there's like in 5 minutes there will be we would continue on the main stage. Is that correct, Cedric, or is this not?
Correct, yeah. So,
okay.
Yeah, in 5 minutes.
Okay.
I would have. Oh, sorry.
Yeah.
I would have one further question, because I was wondering if it is right that electroporation just gives kind of single inserts, because our PIs tend to say that electroporation just is kind of also a good method, because you can integrate a specific DNA fragment several times, so that in the end, for example, the protein production, or general the gene expression is higher, and that this would be a disadvantage of electroporation, but I don't know if it's true.
I don't believe this actually. So I don't know if he has data for this, but like I haven't seen any data on this, and I think electroporation is just the way of DNA getting in there, and it's never just one molecule. And the mechanism of non-homologous end joining, which is integrating something in the genome of the nucleus, is actually not affected by the electroporation at all. So the DNA gets in there, and then Clammy has to do the rest, right? Or the rest of the job. And without that, there wouldn't be any insertion. And this also just happens way after the actual electroporation. So I think if he would be true, then the theory must be that like a single DNA molecule would get in there with electroporation, and with any other method it would be several. And I don't believe this, and that's why I would say it's probably the same to any other method. But maybe someone has data on this, but this is just my theory.
Yeah, but that's also what I thought about it. So
Yeah.
Maybe I ask him.
Yeah. I would like to hear the answer of this question, actually.
Okay.
Yeah. But anyway, I'm way more working on the chloroplast, so I don't have this problem in the end. Very fortunately, I really like to have this specific integration. Yeah.
Okay. Thank you that you take your time to give here this Algae talk and to inform us about how you are working with Chlamydomonas and other algae. And yeah, it was really nice.
Also, thank you for you two that you joined the event for like the first introduction talk. I think like the event up till now was quite nice. I was very happy with that. And yeah, I also hope that other teams can learn from it. And if you need anything, there's like Slack where you can always contact me, or like write in a Q&A section. And don't hesitate to write in there because you think you can just handle it yourself or handle it with just your PI or your supervisor. I think like having many brains working on these problems, on these maybe very easy problems, is always better. So I think that's why this collaboration or the whole idea of this Photoshop community is really great. And yeah, I really hope that we can kickstart this with that.